our students are shy, so nobody will switch on the mic to answer me. So it's okay. You can have the answer in your heart for the questions, or just very simple questions, and you can cross-check with the answers when I review them later, okay? Right, so the first question for today is, are antibiotics, well, antibiotics are powerful medicines that help to fight viruses, bacteria, or microbes or germs? Right, so very easy question, right? The most suitable answer will be bacteria, because antibiotics are medicines only targeting the cellular processes or cellular components of bacteria. So as I think it was mentioned in the videos that we had a look just now before we started the meeting, right? Antibiotics only target bacteria, so we are not supposed to use them for viral diseases. So the second question, right? Antibiotic resistance happens when my body becomes resistant to antibiotics. True or false? Right. So what do you mean? Right? So the answer is false, right? Because antibiotic resistance happens when bacteria becomes resistant to antibiotics. The bacteria becomes resistant. It's not the humans that become resistant, okay? Right, so just some questions to get the session warmed up, right? Okay, so antimicrobials are medicines that you can further categorize them into either antibiotics, antivirals, antiparasitic agents, or antifungals, right? And the hashtag AMR, currently we are using it for antimicrobial resistance, though for the early parts, for now, the usual, when we mention AMR, it would be more towards antibiotics, antibiotic resistance, okay? Right. So what is AMR? I think we sort of like, understand that it's a condition where the killing or inhibitory effects of antimicrobial agents, right, towards microorganism is lost, right? So the pathogens can grow and propagate even in the presence of these drugs, right? So how does AMR occur? Right, some bacteria, yes, they are intrinsically resistant to antibiotics already, but for the other bacteria, AMR occurs when they have this prolonged exposure to antibiotics, right? So usually is when they acquire mutations, right, in their chromosomes, or acquire what we call horizontal gene transfer of antibiotic resistant genes, right? So with that, when they occur, when they acquire these accessories of horizontal gene transfer of AMR genes or mutations, that's when the resistance will occur, and they will be able to grow when exposed to antibiotics supposed to kill them in the first place, right? Okay, and that happens in the bacterial cellular level, right? So we humans are further helping the development of AMR, right? By, as what this comic here is showing, like what Batman says, when we help AMR to develop when we take antibiotics for viral diseases such as common cold, right, or influenza, right? And also because antibiotics are used in livestock farming, right? So with the usage of it in agriculture, in crops development, right, there will be the spillage of antibiotic and these AMR genes, right, into the environment. Okay, so on the other hand, you also have this development void of the antibiotic industry, right? You can see that after the 1970s, right, we do not have any more.
Antibiotics have saved millions of lives since the 1940s when they were first introduced. They conditions like pneumonia.
Okay, shall I continue? Okay, so you can see after. No problem, it's okay, right? So I'll continue, right? So after the 1970s, there's this development void in the antibiotic pipeline or antibiotic industry. So you can see comparing to the years before that, we have this golden era of antibiotic discovery where many classes of antibiotics were developed. But after that, we can see that we do not have a lot of antibiotics that are being developed now, right? So on one hand, you have bacteria learning how to become resistant to the older or antibiotic, the currently available antibiotics. And on the other hand, you have this, development, the drying of the antibiotic lines. So what happens, right, when one day, when we do not have any efficacious or effective antibiotics anymore, right? These 3 photos show, simple procedures. I mean, basic medical procedures that that occurs daily in our hospitals, right? Childbirth, especially by cesarean section, right? Tooth extraction, surgeries, right? So with antibiotics as a safety net, all basic medical procedures will be able to go on safely, right? So in case of any infections, antibiotic is there to rescue the patients from infections. However, with the problem of AMR, and if let's say one day we do not have any effective antibiotics anymore, all these life-saving, all these basic medical procedures would not be able to carry on anymore. And actually our daily life will also be very much affected, right? So perhaps you will think twice before, taking the bus or, going for a walk, because who knows if you get into an automobile accident, car crash, right? So if there's any serious injury, it can be deadly, especially without antibiotics, right? So in the context of also the Sustainable Development Goals by the United Nations, a world without antibiotics, right, affects our health and well-being, right? So it will also, similar like COVID, right, it will affect all the other areas of life, right? Education, work, economic growth, and there will be hunger because we have not really learned how to, produce enough food, right? Livestock farming without antibiotics, and of course there will be inequalities in society, right, without antibiotics. Right. So it's very important that, every one of us, including the public, and especially for you, young microbiologists, right, to join in this effort together to, slow down the pandemic of the silent pandemic of antimicrobial resistance of AMR. And to fight AMR, right, we have to know how to detect it, right, because you can't really fight something that you can't see, right? So how do we detect AMR? Right. So before we go into the detection methods, right? Have you ever thought about how we categorize bacteria or pathogens into susceptible or resistant categories? Right. So just to give an easy explanation, right? Let's say for Staphylococcus aureus, methicillin-resistant Staphylococcus aureus, and let's say resistant towards vancomycin, right? So international laboratories will test many isolates of this MRSA, right? And then towards vancomycin, right? Various concentration of vancomycin, and then they will say that, oh, okay, so most of our MRSAs have this susceptibility towards vancomycin concentrations of less than 1 microgram per mil, for example, right? So 1 microgram per mil will be the break point, right, for susceptible methicillin-resistant Staph aureus, right, towards vancomycin, right? And those isolates which have the MIC breakpoint, or where they can grow beyond the vancomycin concentration of, let's say, more than 1 microgram per mil, will be categorized as resistant, for example, right? So that's roughly how we categorize resistant and susceptible breakpoints for susceptibility testing of pathogens. And we have 2 standards, I would say, breakpoints that we usually refer to, right? Which is, one is the CLSI, the Clinical Laboratory Standards Institute, which is used in Malaysia. Perhaps I think in Philippines you also use this one. So many European countries, they will use the UCAS, right? The European Committee on Antimicrobial Susceptibility Testing, right? So 2 types of breakpoints, right, that are usually used. Right, so now we know how it's being categorized, into susceptible and resistant, right? And then for that, so we want to know, right, how do we then further see to test, whether certain bacteria can grow in different types of concentration, which type of concentration of antibiotics that they can grow in, right? Because we said that resistant means that pathogen can grow in when they can grow in antibiotics, or more, I think a more correct definition will be like when they grow in certain levels of certain concentrations of antibiotic, right? So I introduced to you the first type of phenotype AMR detection methods, which is the phenotypical determination of resistance. I think perhaps maybe some of you in the microbiology department have already done this, maybe in your laboratory sessions, right? What we call the tube dilution method. So what you do is you prepare a series of antibiotics concentration, right? So let's say from here is from 0 until 8, right? Ascending concentrations, and you inoculate the tested bacteria, the bacteria that you want to test, into all these test tubes, right? And then you let them incubate for maybe incubate overnight. Usually what we do is incubate overnight, or let's say 16 to 24 hours, right? According to the UCAS, just now we said the UCAS or CLSI breakpoints that you refer to, right? So after that, you will see the next day, you'll see the bacteria will grow, of course, in the tubes without antibiotics, right? And then you detect the tube which the bacteria is not growing. Let's say this is this tube, right, with 2 microgram per mil. So this will be the MIC of the bacteria that you are testing towards this particular antibiotic, right? So if you further subculture the bacteria from here into drug-free medium, right, and then incubate it another day, right, for the concentration that shows total killing of the bacteria will be what we call the MBC, the minimum bactericidal concentration. Just now for the tube, if you see with your naked eyes, for the tube that doesn't show any growth, that will be the minimum inhibitory concentration, MIC. For the plates, right, that shows complete killing of bacteria, right, that will be the minimum bactericidal concentration, right? So this is what I did also during my university undergraduate. And then when I joined the University for my PhD, let me see, the page is not working. Oops. Sorry about that. Okay, so I think it's this one.
Okay, ma'am, take your time.
Thank you. So when I joined Juntendo University for my PhD, right, we test many bacterial strains towards a variety of antibiotics. So it's kind of difficult if you imagine, right, you want to use setting to test on many strains of bacteria and many antibiotics. So you have to use a lot of tubes and a lot of plates, right? So what I did that time, and this was my first exposure towards the agar dilution and also the broth micro dilution methods in Juntendo University, right? So basically the concept is the same, right? You prepare a media, it can be agar medium or broth medium, right? Containing various concentrations of antibiotics, and then you inoculate the tested bacteria and you incubate it for 1 day. And then when the next day you check, okay, for the strains, you know, that the strains that didn't, for the concentrations that didn't show any bacterial growth, that will be the minimum inhibitory concentration for that particular bacterial strain towards that tested antibiotic, right? So for this, the antibiotic killing breakpoints, the concentration will be known, right? And then for hospital, especially for example, for the hospital where I'm working, affiliated with my university now. So what we do is we have this we use this diffusion test, right? So what the principle of test is, the antibiotics are incorporated into the disc, right? And it can be round shape, right, or in antibiotic strips, right? So when you have this plate spreaded with the tested bacteria, and you incubate it for a night, and then because the bacteria will not grow close to the will be inhibited by the antibiotic killing. So you can see that for if the bacteria is susceptible towards the tested antibiotic, there will be this inhibition zone, right? It will not grow close to the antibiotic already diffused in the media, right? So and if it's resistant, it will grow like, yeah, there'll be overgrowth even onto the antibiotic disc, right? So for the distribution, this will not give you the MIC values, right? So like in our hospital, we will only know that, okay, this bacteria is resistant, intermediate, or susceptible towards this tested antibiotic. Whereas if you use the e-test, right, this is where the antibiotic strips have been inoculated with different antibiotic concentrations, right? So you will be able to get the MIC breakpoints, right, from the test itself. So you can see that for this one, the MIC value will be 1 microgram per mil for this particular tested bacteria, right? So all these are what we call the phenotypical determination of resistance, right? And, for large laboratories, right? So automation for MIC is a must, right? So you have companies such as the bioMérieux, they have what we call like this MIC carts, right? So you inoculate the bacteria that you want to test into the MIC carts, right? Different wells for different bacteria, right? And then what you do is then you pop the carts into the machine, right? So the bacteria will grow, or it will be killed by the antibiotic being tested, right? And after that, you will be able to get the result of the MIC test for the tested bacteria. Okay, right. So that is what we call the phenotypical determination of AMR, right? So it's very much the gold standard of antibiotic susceptibility testing. It's been around for a long time. It's economical. Automation is available for labs which can afford it, right? So antibiotic concentration that kills the pathogen are known, right? Except for the distribution that you will not know the antibiotic concentration, but you can see with your eyes, right, the bacteria growing after you incubate them for 1 day, right? So for this particular test, pathogen culture will be required, right? So your technician or laboratory scientists will need to be able to know how to culture pathogens, right? And then they will need to have experience from reading the results on the phenotypic test, right? And then the thing is with phenotypical determination, you need to let the bacteria grow overnight, right? At least, I think, 16 hours for most of the bacteria. Some bacteria we need to culture longer, right? We'll get to that later. So with like 24, you need like 24 hours to know whether a bacteria is susceptible or resistant to the tested antibiotic, right? So for fastidious pathogens, right, certain bacteria, not all bacteria are made equally, right? So it's not all are E. coli that can grow in like 2 to 3 hours. You can see turbidity in the broth that you're growing, right? Certain, most bacteria will take longer, right? And bacteria such as TB, right, 4 weeks, right? And for those of you who likes to work with fungus, right? Fungus also take a long time, right? So for fastidious pathogens, to wait for them to grow out to let you know the determination, to let you know the AMR value, the AMR status, right? The MIC values of these pathogens will take a long turnaround time, what we call turnaround time, right? And then furthermore, what about for pathogens which are unculturable, right? So that means you won't be able to know the resistance towards antibiotics that you want to use for these pathogens, right? So I would like to also speak a little bit about using the MALDI-TOF mass spectrometry for AMR detection. So maybe some of you have read about this before. So in recent years, there's this technique using the mass spectrometry for bacterial species identification. Basically, the principle of test will be because different species of bacteria will have different mass to charge ratio, right? That is detected by the mass spectrometry. So you'll be able to use the mass spec, right? It looks something like this, right? Expensive equipment, so usually only the bigger labs can afford it. I think our bigger hospitals in Malaysia, the health ministry hospitals also have acquired this, right? So they can this equipment will be able to differentiate the species of the bacteria, so you can identify the bacteria. Oh, is this E. coli? Is this Staphylococcus aureus, right? Is this Pseudomonas, right? And then some scientists are saying that, okay, maybe with that you can also use this. The cursor doesn't seem to be working again. Right. Yeah. So you can also use the mass spec to determine AMR, right? So because for bacteria that has acquired this extra piece of gene which gives them resistance, they will be heavier, right? So the mass to charge ratio will be different. So you can use the mass spec. Most probably you can use the mass spec also to identify differentiate between resistant and susceptible bacteria, right? So there are research on that approach, right? However, a few labs has also been how to say the feedback from a few labs also said that it's not that straightforward as it seems. So I think we will need a few more years of development and also optimization before we can see multi-top being used to further identify resistance in the pathogens that we are testing. Right. Okay. So, but then for the multi-top, usually the pathogen culture is still required, right? So again, this leads us back to our question, right? What about the fastidious pathogens? What about your Mycobacterium tuberculosis, right, which refuse to grow in one day, right? So what about the unculturable pathogens, right? So this question. Will we be able to, directly detect AMR genes, right? That is prevalent in these AMR pathogens, right? So without pathogen culture, since we say that, right, for resistance, right, bacteria will need to acquire mutations, right, or acquire accessory, like acquire extra genes which give them the ability to resist to the killing effects of antibiotics, right? So this is most probably we can do this, and in fact for research, of course, we have been doing this in research labs. For diagnostics lab in the hospital, our hospital has not taken on this yet, and I'm trying working with the the antimicrobial stewardship team and also the clinical microbiologists to sort of have this for our hospital, right? So what we call the NAATS, the nucleic acid amplification test for AMR gene detection, right? So the genes that causes AMR detection, many genes have been discovered and reported. So you know the genes and you know the gene sequences. So right, you can design the primers and then use that to sorry about this again, right? And use that to detect the absence or presence of AMR genes, right, in the bacteria that you are testing. So, right, so if you have worked with PCR before, right, the amplification of the genes, right, of the AMR genes, right, absence or presence of these bands, right, will tell you whether the bacteria is carrying the AMR gene and whether, most probably, if they're carrying this particular AMR gene, most probably this antibiotic, you can't give it to the patient, right? So, and then with times of COVID, almost every hospital lab now has the real-time PCR. So, right, a version of the PCR where you can even see the amplification process going on, right? So with that, we think that the NAETs, right, for the AMR gene detection, probably we can see this phase being implemented in hospitals in post-COVID. Most probably this can be the acceptance of the clinical microbiologists and also the physicians towards this maybe hopefully will be increased after COVID, right? So some points about using NAATs, right? So the AMR gene sequence must be known, right, for the that you want to test, right? And then your technicians and your lab scientists will now have to have another skill, right? But just now we said that for the phenotypical resistance, they will need to know how to culture bacteria, right? Anaerobic bacteria, right? Fastidious bacteria, they will need to culture those bacteria. Then your technicians will need to know, let's say if future microbiologists, maybe you have your own lab next time, right? So your technicians will need to know how to do DNA extraction, PCR and gel electrophoresis, or even real-time PCR, right? And then the exciting possibility of detecting pathogen without culturing. Yes, right? Maybe if you have enough DNA of the pathogen inside your samples, right? It's possible to do that. And with that, you can cut short the turnaround time because, right, DNA extraction and PCR gel electrophoresis that will take you maybe 4 hours, right, for the results, right? So compared to the overnight cultures, right, to get the antibiotic susceptibility breakpoints, right? So 4 hours, 5 hours, you can let your doctor know, okay, can this antibiotic be used or not, right? And well, for PCR reagents, and a PCR machine is now it's a common equipment, so the cost wouldn't be too far apart. Wouldn't be such a big hike in the expenditure of the lab, right? But the retraining of the technicians is one thing we need to consider, right? And then on the whole, the PCR equipment is definitely cheaper than the mass spec. Remember the mass spec we mentioned just now, and easier to maintain, right? Cheaper than the multi-TOF, right? So, but the few things that we need to ponder is, does AMR gene carriage improve phenotype resistance, right? Because in some instances, let's say if there's mutations to the AMR gene, right? So the phenotype might not come out, right? So you might not, so we just not say that this NAATs is for the detection of absence or presence of AMR gene. So if the resistance is caused by mutations, let's say for vancomycin intermediate Staph aureus, right, you might don't think you can now, until now we can't really detect that using a PCR technology, right? And the MIC values are not known, okay? Right, so just now we talked about the fastidious culture, right, like tuberculosis, right? So we using NAATs, right, like this platform, the Gene Expert from the Cepheid company, right? We call they call it the Expert MTB XDR platform, right? So what you do for this is a revolution, right? The patient comes, right, and then have been coughing for a few months, right? High fever, loss of weight, night sweats, right? So sort of like you're thinking like maybe it's TB, right? And then in the prior to, if you use phenotypic culture to detect that, you will need to take a sputum sample and culture it for 8 weeks, and then you know that that's TB, and then furthermore, like you need to culture it again to know the antibiotic resistance of the TB isolate, right? So for this Gene Expert platform, the patient comes and then give you a sputum sample, right? And then you insert it into this cartridge, right? And then you pop into the Gene Expert platform, right? This magic machine, right? Where the DNA extraction, the PCR is carried out inside the machine itself. And then within 90 minutes, right, you can get the information, right, whether your patient has TB or not, and if it has this rifampicin resistance or not, because this will sort of, right, divide your TB into the high risk XDR TB or the MDR TB, right? So within 90 minutes, so it's the same day, sort of like same day you can optimize treatment for the patient, right? That's very, very nice, right? You don't need to your patient go back into the population. Imagine right now, COVID patients, when we know them, we will want them to isolate, right? Imagine TB patients prior to this, right? You ask them to isolate, but do they really isolate, right? So spreading the TB maybe in commuter trains, right, in school or work, if they go to school or work, right? So this will revolution. This has been used in some hospitals. It's a very expensive, well, very expensive for my hospital, right? But some hospitals have used it because you can give treatment earlier, right? So you can detect the pathogen and the AMR at the same time, right? Automation, so reduce risk for your technician being exposed to the TB bacteria, automated, and then yeah, it needs the test is more pricey compared to going in the Lowenstein Johnson, right? So you can also look into this biofile film array system, right? This one is also like you have this magic box, magic cupboard where you just pop in the samples that you want to test for the pathogen, and it can also test, have some limited tests for the antimicrobial resistant markers, right? So walk away, pop in the sample, and then you walk away. So of course, when you have this magic cupboard, magic cabinet, magic equipment, right? It will be costly, and then will be some cost for the hospitals, right? So that also has to be factored in when you choose the method for AMR testing in your future laboratories the next time, right? Okay, so I've been talking more about this clinical bacteriology, right? So it's more for AMR testing in hospital. as I've shown the figure just now, like we do use antibiotics for livestock, and because of that, there's a spillover of the AMR genes to the environment. So now we are very concerned about that, because we know health affects not just humans, right, but also the animals that share this earth with us, right, and then also the environment itself. So with the One Health initiative and approach, I think some of you are in this webinar also from the biology department, right? So maybe you are future botanists or zoologists, right? And you work with animals or work with plants, right? So testing of AMR genes, right? The presence of AMR genes in these different environments are also something that is gaining importance, right? And then also we're just saying looking outside for the One Health initiative, but actually don't forget to look inside, right? Because the gut flora, right? When we take antibiotics, I'm sure most of us have taken an antibiotic before, right? So when you expose, these different bacteria in our guts, in our colon to antibiotics, right? Some of them will also learn how to become resistant, right? And some will be killed by the killing effect of the antibiotic, right? So there's this notion that we should also, look into our gut flora, gut bacteria inside our gut bacteria body, right? To check what we call the gut resistome, right? What is the reservoir? Is there a reservoir of AMR genes inside your colon also, right? So now to do that, right? You can, of course, you can use the NNATs, right? Or you can use the phenotypic test. For you isolate first. To use the phenotypic test, you need to isolate out, right, different strains and different types of bacteria, right? But if you're working in AMR in an ecosystem, right, and some it has been reported that only 2% of bacteria are culturable. So right in the jungle, in the lake, there are many bacteria that is unculturable. Same as in our colon, there are bacteria that is unculturable. So what you can do is if you still want to check for the AMR genes or AMR reservoir in these environments, you can use gene arrays, right? So you can't, most probably you will not use the method of culturing them, right? But you will use the nucleic acid-based testing, right, such as this AMR gene arrays by QIAGEN, right? So let's say you take the sample from the lake water, right, and then you extract the DNA, and then what you do is you put the DNA in this 96 well plate or 384 well plates that already have primers of the antibiotic resistant genes, right? They're specific for each antibiotic resistant genes in each well. So, and then you do a real-time PCR and it will tell you later, okay, what's in this lake water, right? What are the antibiotic resistant genes available in this can this work? I think some have difficulty going to the previous. Yeah, that's a lot of pipetting work if you want to do this. Like, so imagine 96 well plate and 384 wells. But anyway, we'll solution and we'll see later, right? So what you get is, like just now I was mentioning, you want to check what is the AMR reservoir, AMR genes in let's say lake water sample or wastewater sample, like what is being done in Nigeria here, right? So you can know, right, what are the antibiotic resistant genes in this particular sample, and you can even, because it's if you use the real-time PCR, right, you can even know, right, the percentage of the prevalence of the antibiotic resistant genes, right? Yeah, you might want to sample local birds. So this one was when we do not have gulls in Malaysia, so I gave this slide when I was talking to my students. So if we have some friends from Malaysia here, say like, do you want to sample our crows, because that's what we have here, right? And so you might find antibiotic resistant genes in the environment, right? It's not something that scientists are making the public concerned about, but it has been found, right? Antibiotic resistant genes in the environment. And I'd like to just talk about this resistome map, which I think they have been very active on Twitter. They use the qPCR system, right? And right, if you find that it's a lot of pipetting work, and if your principal investigator have the money, right, you can outsource the determination of antibiotic resistant genes to resistome map. I think they have some robotics to do the pipetting for the analysis, right? So what they do is they determine the AMR genes, right, in various One Health associated various environments in One Health, right? Agriculture, hospital, wastewater, treatment plants, wildlife, right? So the CEO of I think the chief scientist of Resistomap is an Indonesian lady, and I think the company is now based in Helsinki in Finland, right? So if you're interested in this, you can check it out, right? Okay, so go big or go home, right? So when we talk about antibiotic resistance, right? The big brother of determining antibiotic resistance will most probably be using whole genome sequencing, right? Of AMR pathogens, right? With this method, you will be able to know everything, right? You will not just know what are the what species is the bacteria, right? You'll be able to know the AMR gene content, right? You'll be able to know virulence gene content, right? And also other genes that is associated with the biological phenotype of the pathogen, right? So it's not, because it's still expensive, I think this is the next generation sequencing platform. This is a third generation sequencing equipment, right? So this one is kind of, if you are familiar with it, this is the MySeq platform. So it's about, if you have seen the computers with the larger monitors from maybe the 90s. I'm not sure whether if you guys, I think you guys are not born yet, right? So that will be like take the space of the large computers, the computers with large monitors, maybe 2 monitors, right? 2 or 3 monitors, and you can sit on the desktop. For the third generation sequencers, right, you have the large sequencing platform from PacBio. I'm just showing here the Oxford Nanopore equipment, which is as small as a pen drive, right? So you can take that to the field if you need to, you know, work, let's say in the jungles of Malaysia. You want to detect the antibiotic resistant genes there immediately in real time, you can take it to the jungle and do the sequencing there, right? And have the readout there, right? So the technology is still expensive. So maybe around, I would say 20,000 pesos, maybe for us it's 2,000 for one genome, right? So it's not yet for day-to-day diagnosis of AMR, but the company guys have been telling me, telling us, encourage us to try it for sepsis. Now, sepsis is a condition where the bacteria got into the patient's blood, and it can be deadly for the patient, can cause death very quickly, right? If the suitable correct antibiotics are not administered immediately, if the sepsis is caused by bacterial infection. Now, the usage of the nanopore, right? You at once, not at once, about 2 to 3. You just need 2 to 3 hours of the sequencing, right? And then the sequencing is done in real time. The data will come out in real time, so you'll be able to know bit by bit. Oh, so it's resistant to this antibiotic, that antibiotic, as the result comes out. So by 2, maybe 3, 4 hours, you can also know what is the infecting bacteria, and most importantly, what are the antibiotics that can be used or can't be used for the patient. And hopefully this will be a game changer treatment for sepsis patient, right? So for now, the usual usage of whole genome sequencing for AMR detection or detection of AMR pathogens is for genome surveillance for AMR pathogens, right? Find that. Okay, so I'm just gonna do what I'm doing, right? So this is a very nice study from the Philippines, right? And I think the second author of the study, Dr. Melissa, is here together with us in this webinar, right? So for this study, what the authors did is they look into the antibiotic resistant genes and also profiles of 4 important gram-negative bacteria, right? Acinetobacter, K. pneumonia, Pseudomonas aeruginosa, and E. coli. Right. So you can see from the graphs itself, the figures itself, you can see the trends in the changes of the antibiotic resistant profiles, right? So for this particular pink profile, right, you can see that in the beginning, it's in the 2009, right? This particular AMR clade pneumonia, you do not have that, but towards the 2016 and 17, it becomes, you can detect it more in the Philippines, right? This is across a few sentinel sites, right? I think if you have any questions, further questions on this, you can ask, and Dr. Melissa can answer them later, right? During the question and answer time. So they did the whole genome sequencing for 2 years, 2013 and 2014, right? For isolates collected from these 2 years, and with the sequencing, right, you will be able to know more about the resistotypes, the resistant profiles of these isolates. Let's say for clap pneumonia, right, you'll be able to know whether if they are carrying the BLA NDM gene, right, or what NDM plasmids are they carrying, right? So this shows the sentinel sites of the study, the whole genome sequencing study, right, which was performed in Philippines, right? I think Caraga is over here, right? So yeah, and it's a very nice study. So do if you're interested, please do look up this paper, right? And have a look at it, right? So that was in one country. So let's say if and we know that with COVID, we know that infectious diseases doesn't require a passport, right? It doesn't have these limitations to one particular country, right? So what can we do, right? If let's say I want to exchange, let's say I sequence the Staph aureus MRSA from my university, right, National University of Malaysia, and I want to exchange, I want to compare my strains with the resistant profiles from those from the Caraga state, from the MRSA from Caraga state, right? So now I'm working with some collaborators together with a grant from this Public Health Alliance for Genomic Epidemiology. So what they developed this bioinformatic tools called harmonization. So what we have is like, even if, let's say, we are using different sequences in different countries, right, or different AMR tools, right, to analyze your genome, your whole genome samples from different pathogens in different countries, you will be able to use this tool to harmonize them together and make a standardized gene report, right? So you'll be talking like apple to apple and not comparing apple to orange. So you'll be able to have a nice comparison of the AMR genes contained by the pathogens that you want to compare between two different labs. The labs can be, in 2 different states, let's say between Luzon City and between Mindanao, right, and Caraga. And you can also compare between countries, right, between Malaysia and Philippines, right? So you can look into the Resist TB platform. Now, the TB is drug resistance in TB is a very serious problem, and it has been with us for a long time, and the attention towards it is not as much as we like. So that the disease is not really is not in control, right? So with whole genome sequencing of TB strains, right? There's this Resist TB platform, right? You can Google it later and then have a look into it. So scientists working with TB, you can genome sequence your TB isolate and then deposit it in this platform, right? And with that, the global consortium will be able to monitor the changes and trends, right? Using whole genome sequences of these TB strains, right, in the whole world. So same as what has been done with the COVID-19, with the SARS-CoV-2 viruses, right? So with whole genome sequencing, you know that there's this Wuhan strain, which is now no more and replaced by the Alpha strain, and then later now Delta, right? So with the Resist TB also, it monitors the dissemination of different strains of TB, be it multi-drug resistant TB, right? Extremely drug resistant TB, right? So you can check it out here. So the whole genome sequencing is one way for us to, not just to detect resistant genes, right, in the TB, so that you can give the best medicine for your patient, but also to monitor how these pathogens change over time, right? And be sort of prepared, and to sort of can let you have some hypothesis, okay, what will happen later, and maybe design better drugs to control or have better public health policies to control and hopefully eliminate the disease itself. Right. Okay. So we've talked about phenotypic resistance. We've talked about phenotypic detection of AMR, and also using nucleic acid detection of AMR, right, and even whole genome sequencing. So I will just highlight this rapid antibiotic susceptibility testing. I think this is considered the phenotypical testing. It's something which I heard of in 2019 when I attended a conference in Korea, right? So this is also phenotypic testing and observe the growth of bacteria, but it's done in real time, right? Monitor real time of growth of bacteria and tested antibiotics. So it's usage of like very advanced microscopy technology. So you see the bacteria grow in real time, right? Divide in real time. So what you do is you expose this bacteria to, antibiotic that you want to test and you want to see whether they are resistant or not, right? So if they are, can be by the bacteria, so they will not grow. But let's say if they are resistant, you can see the cells multiply in real time. Right? So you can make the call. I don't know, maybe if it's validated, right? So you can make the telephone call to the infectious diseases physician and say, okay, no penicillins for this patient because I've seen the cells, growing in real time, right, when they're exposed to penicillin, right? So a few interesting research have been done on that. It's still concept, right? Prototypes, right? So very short TAT, right? 2 to 4 hours. Maybe, right? So hopefully this will help in accurate and faster antibiotic dispensing for the patients, right? So as I have mentioned, what I've mentioned today is most about the detection of AMR in bacteria, right, towards antibiotics, right? You do have AMR also. We mentioned just now, AMR also consists resistance towards antivirals, antifungals, and antiparasitic agents. So if you're talking about resistant methods detecting AMR towards the other pathogens other than bacteria, the methods are roughly almost the same, right? So it's either phenotypical determination or, you use DNA technology to see whether, the resistant genes are there. Well, for antiparasitic agents, you want to detect resistance in anti-parasitic agents, it can be very interesting, some very different and interesting tests because parasites, you have many types of parasites, right? Worms, amoeba, right? What else? Single cell parasitic agents, right? So malaria parasites, right? So I've found this. So you can also do the detect resistance when you grow them in the other anti-parasitic agents, right? Or detect for antibiotic resistant genes. But you can also do something like check the egg production, right? Like for here, if the egg reduction is less than 90%, right, post-treatment it suggests anti-parasitic resistance. So if you're interested in this, I'm not familiar with this, so I think you can arrange a talk from the parasitologist, right? They will be able to brief you more about resistance to anti-parasitic agents, right? If you're interested. Okay. And then, right, so with COVID-19, right, the most of the attention in public health agencies is towards COVID-19, right? And I do not deny COVID is very important, and it's a pandemic, right? So we need to pay attention to it. But also, I would like, because you guys here are future microbiologists, so I would like to really remind you that AMR is a very important problem, right? It's a silent pandemic, right? So we have the newspapers every day telling us how many people die from COVID, but do we have this AMR deaths in the newspapers every day? How many patients actually die from AMR? We do not have that figure, and actually it has been reported that AMR is actually this pandemic is actually occurring under our nose now. It's just that it's not it doesn't make a noise such as COVID. I will encourage our future microbiologists to also, pay attention to this particular problem, AMR, and yeah, join hands together with us, right, to fight and to slow down this silent, slow motion pandemic, right? Okay. And the WHO has been interested, has been very concerned with the AMR since, I think, before 2015, right? In 2015, they launched the global AMR surveillance systems, right? So if you guys here are very passionate about AMR and you feel that this is your calling, right? So please read up about the GLASS in the internet, right? So it's an international effort from the WHO to monitor AMR trends, right, in different countries, right? So yes, Malaysia and Philippines also submit the AMR data, right, to the GLASS initiative, right? So for now, they are collecting data on resistance, mostly phenotype resistance, right? So in future, they'll also be collecting, encouraging, right, the detection or whole genome surveillance, as I mentioned, whole genome sequencing of AMR pathogens, right? So to have this better surveillance on AMR pathogens, right? So we'll be able to be prepared, right, for the next pandemic before it comes, and hopefully prevent it before it comes. Right, so I think I'm coming to the end of my talk, right? So just some food for thought for today, right? Detection of AMR, ideally it should be rapid, right? So because you want to have case isolation and tracing, right? You do not want your MRSA patient to be, right? You do not want your very friendly MRSA patient to go from, you know, hospital ward to hospital ward to, you know, make friends with other patients and also pass the MRSA to them at the same time, right? And also the detection should be rapid so that you can give the correct antibiotics, right, to patients who need them, right, to prevent the worsening of clinical course, right, and then prevent further resistance, right. And I heard this during a talk, and I think this is during a talk, and I think this is very important. The method of AMR detection should be cheaper than antibiotics, right? So this is to encourage detection, right, before you prescribe antimicrobials. So imagine if, let's say, antibiotics are just 50 pesos, for example, and your AMR detection method is 500 pesos, so right, 10 times more expensive. So doctors will be at the how to say, they will be pressed to, you know, give antibiotics instead of, you know, using the methods to properly detect resistance before prescribing antibiotics, right? So the AMR detection should not be too expensive, right? And it should be cheaper. Ideally, it should be cheaper than antibiotics, right? So this will hopefully prevent further resistance. And then also, what we talk about today is in the in vivo setting, right? In tubes, in plates, right? So it's you know what happens when you take antibiotics, right? It's antibiotics get into our bodies, and then it meets with the our immune system. It meets with the drug binding molecules, drug binding proteins in our body. So right, so it's different. Sorry, what I'm talking about today is in the in vitro setting, right? So what happens in the real in vivo setting when antibiotics, right? When antibiotics meet the bacteria in our body, right, or in livestock, right? So what really happens there, right? So we still do not really know the complete clear picture about this, and I think it's also worth investigating, right, for our future microbiologists, right? So my message for today, right, take-home message for today: aim no more in the dark, right? Future microbiologists, let's all work to develop affordable and rapid AMR diagnostics, right? Because that's very important to control AMR, right?